TSS or translation start site, they also provide important signal as far as the gene prediction is concerned. Translation starts with ATG that codes for methionine in a polypeptide. Here in these examples, we see that we have those TSS in the middle. It's actually a file where the sequences are aligned with one another, and we can look into their ATGs, and we see there are some neighbors which are around them. If we closely look, we see that around ATG we have C's and G's most frequently present in this example. We might want to use this thing in order to identify the presence of these these TSS. So we assume that certain nucleotides, they prefer to be near or being the neighbors of those TSS, and these bias distributions then they can be informative. So while looking into the frequency of those particular nucleotides around TSS can help us support the idea that those TSS are the real ones. For this, what we can do here is, since these TSS, they are present as a triplet codons, and we can take hexamer frequencies from all those sequences, and then on those hexamer frequencies we can observe which hexamers are present around those particular TSS. So this is how we do that. So we can calculate the frequency of those coding versus non-coding regions, and we can get the positions or individual nucleotides with this equation. So that here we say that Fi, which is the frequency over some x, any nucleotide, is given by sum of the frequency of their log of the ratio of their counts in that particular position divided by total number of nucleotides. So C is the counts or presence of those particular nucleotide divided by total. So actually that will give us the idea of the frequency. So when we take the log, we can put it into this form, which we call it as information. So in this way we can have their frequencies. So based upon the equation which we have seen in the previous slide, we can come up with a frequency table about the frequency of the neighbors around this ATG. We have ATG in the middle, and we have their neighbors. Before this ATG, we have those numbers indicating what was before that, and we have -1, -2, and -3 and -4 locations, and same way we have plus numbers, so that indicates the positions which are we call them downstream positions. If we look into -4, we see there are more C's as compared to rest of them. Position number 3, A and G somewhat comparable, and then we have C's here. If we look into +3, we have G's, C, and again G's here. So this is how we can come up with the frequency of some nucleotides around these start sites. So there are some nucleotides which prefer more to be there as compared to some of them which prefer not to be there. So if you are getting these kind of frequencies, and you are getting some ATG, you might support your point that this is the true start site, or while looking into these frequencies you can expect that you can have a start site over there. So let's do an activity in order to elaborate this point. So we have two sequences. One of them is CACC ATAGC, and there is another one where we have ATG and we have some neighbors, the green one and red one. So how can we say that one of them belongs to a place where we have this TSS? We can use frequency table as in the scoring function. So the frequency table gives us the frequencies of those nucleotides around which was actually calculated from some data or some training data. We have seen that in the previous slides. So we can get that frequency, and then we can divide it by the expected frequency, that is 0.25, since there are 4 nucleotides. So the frequency from frequency table divided by the expected frequency, that gives us an estimate of which neighbor is a strong neighbor or which neighbor is a poor neighbor or not that much strong. So we put it together in this equation, which is called as a scoring function or S of I, where we are summing up for the frequency of whatever those segments are there or whatever those nucleotides are there. We take their frequency from frequency table and we divide it by 0.25. And then we convert it into the log scale. We want to play with big numbers. We can call this equation as the information content. So here is our frequency table. We pick the frequencies from here and we put it into these whole numbers together. So we add all those, we calculate those log ratios. We get their frequencies from frequency table and we divide by the expected ones. So in this way, in the end, we get a positive number. It is 13.69 on this side. Let's see what about the other sequence. Over here, it's 9.44. So which one is the stronger evidence? So in our case, we'll prefer the ones which are having the highest value or more value. So probably the green one. Let's look into this algorithm, which we can put it together with whatever so far we have learned into this as far as this section was concerned. So we can build a mathematical model based upon collected translation start sequences, and for each sequences we apply the model and get a score. So we get those frequencies and we divide with the expected. In this way we came up with a model. And then if the score is larger than 0, it is we can predict that there is a translation start site. Higher the score, higher the probability that the prediction is true. So TSS prediction can be an important step in gene prediction, and it can be predicted by observing or by using the frequency of the neighboring regions.